and welcome to Scope, where we're making a splash with an episode all about aquatic science. Let's dive right in by taking a look at some underwater beauty.
Native freshwater fish are always on the move, looking for food and shelter and avoiding predators.
But unfortunately, there are lots of man-made structures in our waterways that are blocking their way. Hi, I'm Harriet. I'm a researcher here at the University of Queensland's Eco Lab, and we're looking at ways to help our fish get from A to B.
A big part of the problem are culverts, pipes built under roads for waterways to flow. They're important to help avoid flooding events, but culverts change the way water flows, making it rush through the pipes faster than normal.
And that's bad news for our freshwater fish species. Most of them are under 10 centimetres long and aren't strong enough to swim against the fast flows to get where they need to go.
To improve these culverts, we need to study how fish behave inside them. In our hydrodynamics lab, we built these long tanks called flumes, which replicate the culverts. We can observe how fish use the flumes, then we can design ways to adapt the culverts so wild fish can use them more naturally.
One design are these long beams that we place along one side of the flume. This shape changes the way water flows.
It creates what we call a reduced velocity zone. It means that water close to the beam flows slower than the water in the centre. We've learned that fish swim close to the beam, avoiding the fast water flow to save energy. Beams like these could be fixed into real culverts to help fish swim through easily.
Water flow is just one of the problems we're trying to solve. Some culverts can be more than 50 metres long, which means they get dark, and not all fish swim in the dark.
We've built a blacked out flume to research which freshwater fish prefer daytime conditions. Surprisingly, not much is known about this. We're now learning which species are less active in the dark and working on ideas to help them, like culverts with skylights to let daylight in.
To help collect all of this species data, we've turned to some tiny technology.
These small tags work like microchips in a pet dog or cat. With a few drops of anaesthetic and a small incision in their belly, we can tag a fish in no time.
We can use a handheld tag reader like this one to pick up the tag in a fish as the fish swims past. Then this data is sent to our computer where we can analyse how each species is behaving.
This means we can release dozens of different tagged native fish into our flume and gather lots of data all at once to understand how each species copes with the water flow and changes in light availability.
So thanks to our research, native fish like these could soon be swimming much more easily in the wild.